Okay, recording's in progress. All right. Well, Jim says she's coming. I'll keep an eye on that. But let's
Coming.
admit so many things at the same time. Let's start.
You need to share screen.
Yeah.
Um,
And then hang on. I need to go back to Zoom and share screen. Wow, where is my Zoom? Go here, go more, go share screen. Let's share desktop number 2. Okay, cool. I think it's working. Maybe can someone close the door back there? Just in case. This name. And you know what? Let's get rid of this big thing up top. Let's put, where's the mouse? Let's bring it over here. Okay. So, konnichiwa. Let's talk a little bit today. So there's some people who are absent today. I wanted to talk about species and describing from today, but there's 3 or 4 people that are absent that I really want to see the species part for describing species. So today I've switched the order, but this makes more sense, actually, thinking about it. Last week we talked about notebooks, we talked about data, backup, we talked about things like that. So the next thing, logically, is going to the ocean and collecting specimens and or maybe working in museums. So today I want to talk about collecting specimens and museum work. This collecting part, I gave a class last year in summer during lab meeting very quickly. So some of you have seen part of this, but I've changed some parts of it. And this part is brand new. So this is what I want to talk about today, probably for about 1 hour. So last year, Fiona and Kristen, I talked about collecting specimens, right? And I remember I said, it's easy, right? Collecting specimens is easy, right? And then I put, well, there's actually a lot of things that you need to think about, even when you're diving, unfortunately, or snorkeling. And actually, if you think about it really brutally, when you're collecting, you're killing animals, right? And so I like collecting. I don't like killing animals, but I like collecting. I like going and trying to find things. Most of us work on colonial animals, but not all of us. So you're actually, but if you think about it this way, you're killing animals. So not only is there permits or ethics, but you really need to do things properly. Otherwise, you're just killing animals. So if you collect properly and you do it for science, you know, this is good, this is fine. But you always need to consider permits and ethics. And I'm going to talk later, a little bit later in this class about permits. Okay. But I think more from an ethical point of view, don't forget this. As sad or as awful as it is, please don't forget this. Okay. The other thing is not only from ethics point of view, but also from logical or logistical point of view. Now you may not be able to send specimens to other countries often. You may not be able to receive specimens from other countries. You may not be able to publish without proper permits. When you go to submit a paper, they will ask, journals ask you for your permits or a copy of your permit. So if you have a permit, great. And in some cases, for example, in Okinawa, we do not need a permit for nematodes. We do not need a permit for polychaetes. We do not need a permit for zoantharians. But we do need permits for corals. We do need permits for sea cucumbers. So it's case by case. But if you don't have a permit, you could get into trouble. So, and you will never publish it. As well, Recently, in the last 5 years or so, there's been a lot of talk about the Nagoya Protocol. This was made at a meeting in Nagoya, hence the name Nagoya Protocol. This is about biodiversity and sharing and ethics consideration. Do any of you know the Nagoya Protocol? Yeah, Nuria does. Anyone else? Stefan does. So the basics of the Nagoya Protocol are the rule was made to prevent helicopter science. Helicopter science is when a researcher comes from one country, lands in another country, collects an animal, goes back to their home country, never talks to the first country, never talks to researchers, maybe never gets permits, and publishes papers. That's helicopter science. So as much as possible, if you are doing work in another country, you want to work with local scientists, you want to collaborate, but you also need permits. And so Nagoya Protocol, what the Nagoya Protocol does is, if we go, for example, if we go to Reunion, for example, let's say we go to visit Stefan's homeland, and we collect, and we don't involve anyone from Reunion, not even Stefan, and we collect things, we bring them back to Japan, we publish a paper. If someone in Reunion sees our paper and says, hey, that's wrong, they can contact the Japanese authorities. The Japanese authorities investigate, and if we are in trouble, we go to jail in Japan under Reunion rules, or we pay a fine to Japan based on how much the union says we pay. So in other words, it's like an extra boundary situation. So you have to be very careful with international research, basically. So how do you avoid international trouble? Number one, you work with people in that country. So if we go to the union and Stefan's there, we work with Stefan. But also we make sure that Stefan knows the rules, right? If Stefan doesn't know the rules, it's no good. So you work with people in the other countries, but you also make sure you have permits and you know the rules. It's really, really pretty serious. So that's number one. The other issue is CITES. This is the Convention on the International Trade of Endangered Species, CITES. This affects anyone working on not only elephants and pandas, anyone working on hard coral, Scleractinia, anyone working on black coral, Antipatheria, anyone working on blue coral and relatives, Heloporidae. A lot of these groups are CITES. And so CITES has similar rules to Nagoya, where there's permits and jail time and fines and strict, but it's above and beyond Nagoya. So if you have hard coral from a country and you have no local collaboration and no permits, you could get in trouble for both of these, which is really, really bad news. So especially for anyone working on hard coral, black coral, blue coral, for anything international, there's extra paperwork. For example, we still don't know if we can send specimens from Japan to other countries for CITES. Right now, Kairi and Ayaka are working on it. It's huge amounts of paperwork. It's really troublesome. Yeah.
CITES has like international rules, or Japan has CITES rules, and US.
CITES and Nagoya Protocol are both international rules, but for example, within CITES, each country has rules for how they do the paperwork. And Japan, which loves paperwork, and CITES, which is already a lot of paperwork, it's tough. So yes, just things to remember. That's why for some things, it's much safer and much easier to work domestic. The problem is, domestic is only one part of the world. Species don't care about boundaries, right? So if we want to understand species internationally, we need to work internationally. But these are things that you really need to be aware of. Okay. Before I go to the next one, questions? It's okay? Okay. So when you're sampling, let's say we go sampling, yay, we're going to the ocean, right? For some people, it's really easy. Like Mari goes and collects sand, right? But for other people, it's a bit more difficult. But as much as possible, even if you're Mari, of course, you want good in situ images. You want a picture or a record of the environment. You know, maybe it was cloudy, maybe it was rainy, maybe there were waves. It's good to have as much information as you can. If you're looking at an animal, maybe you want a ruler or something of size underwater, a knife or a flashlight, anything that you know the size of. You might want images of the surrounding environment. I do this often when I find something unusual. Not only do I take a picture of the animal, but I try to take a picture of what the reef looked like or what the area looked like. Your camera will look at the time. You have your depth on your dive computer, so you could take a picture of your dive computer. But all of this information could become important later. In other words, this is your only chance. And I think you guys are better than me. I grew up in the age of film camera. So you only had 36 photos to take and you were finished. Do I take a picture? Yes, no, it's okay. With digital camera and SD cards, you should not ever worry. You should be taking hundreds and hundreds of photos, right? No problem. As well, you know, make sure you know where you put your specimen. You put it in a Ziploc with a number or something. Because really, this is the thing. This is your only chance to be underwater. You can look at your photos later, you can look at your specimens later, but when you're there in the field, whether it's reef walking or diving or on the boat, cruise or whatever, that time is your only chance to get that information. So don't waste it. Work, if you think, oh, I'm tired, work hard now for 5 more minutes, then get the latte after, right? You're already thinking, right? Okay. I talked about labels last week, right? So this is kind of a review. I won't say too much about it. Data, labels, information, all of that though should be organized the same day as much as you can. This is tough. If you've done 3 dives or you're up late on a boat and you're like, oh, I want to go to sleep. You forget very quickly. Oh, you will forget. It's amazing how quickly you forget information. So please write things down. In other words, if you really forget to get information, your specimen is useless or it's not as useful. If you have no information, it's useless. If you don't remember the date or the depth, it's okay, but it's not great. If you don't remember the location, it's really bad. So basically, to show whatever you've collected some respect, try to write down that information, right? That's sort of how I think about it. So what do? Write down all your information in your notebook, right? Everyone's got a note today. This is amazing. So write your information in your notebook. If you write your images or information or something, I write my photo file numbers, right? Excel file, but also in your notebook. Make sure everything's linked. I put mine into an Excel file. I make labels after this, the same day. So I have my notes on my specimens. I have an Excel file. I look at those 2, and then I write my labels, and then I put my labels in my animals. Labels. This is what I want to talk about a little bit. Okay, I talked about numbers, idiot proof, for example, like that. This means James Davis Reimer, 2022, July 27th, dive 2, specimen number 19. This is just my system. You can have your own system, but make it a system that you understand that's easy for, I can explain this to other people very quickly. Okay, but there's a lot of things to think about with labels. What is needed? Species name or ID, right? Well, I'll talk more about this in another class, identification. Some kind of scientific name. If you're not sure, we'll talk about this in another class. AF. AF means affinity. It means you think it is close to this species, but it's not that species. CF means you think it is this species and it probably is this species. Confers with. これと近いだけどそうではない。その種ではないけど近い。この種だろうは CF, right? Collection dates, time, you don't necessarily need time, but some people put it in if you're doing something like that. Location, also make sure your spelling of your location name is correct. If you're not sure, there's a lot of problems with Japanese specimens in the past because people couldn't speak Japanese. And the writing is like, is this location or this location? If you're not sure, get someone to write kanji even. Even better, GPS, latitude, longitude. Maybe also location, coral reef slope at GPS point, 9090. That's good, right? Collector's name or the person who ID'd as well, important. Depth, preservative, fixative, right? Ethanol or formalin, what percent, RNA later. Well, usually not RNA later because it's genomic. But also how you make your label, waterproof paper, good waterproof paper, not cheap waterproof paper, good waterproof paper, N pencil, because it will not go away with ethanol, and labels are inside the specimen. If you are doing genomics and you are worried, wear gloves, or you can put the specimen outside, the label outside, but there are very high chance the label goes away. It's way safer to put things inside. If you write outside with pen and ethanol spills, the label disappears. Yeah, no, you can't, but you need to think of if this is like the standard and you're working with something different, you need to think of a way to make sure no matter what happens, you know what your specimens are.
Yeah, it's just inside.
Yeah.
I can't read it. I score in the mud.
No. So I understand with genomics, this really changes. And we've done genomic analyses of museum specimens. Like 80% of the DNA is human after 10 or 15 years, because people keep touching the labels. So, gloves, right? There's ways to think about this, but some people use barcode machines, but then the barcode sticker can get old or broken. The only safe way to know for sure is this, but this is also bad for genomics. So you might need to maybe do subsample. One sample with label, half of the sample, no label, safe, right? For each, but adapt to that. But what I mean by this mainly is a number on the outside of a jar alone is probably not enough. That's what I'm just warning you on that. Questions or comments on this part? Easy? Cool, okay. Long-term storage, you should imagine that your specimen someday, your specimen will end up in a museum. Hopefully, because this is the other thing, you never know how important your specimens might become. For example, when I went to Ogasawara in 2007, I was like Koida sensei now. I was a brand new sensei. I went to Ogasawara 2007, maybe Kairi was like elementary school or something, right? And I collected an anemone. Turns out it was a totally new species. Izumi-san described it last year. But I didn't know, I didn't think it was important. I was just like, an anemone. And I just wrote anemone, pickle, put in a jar. 12 years later, someone comes along, wow, it's amazing, right? So you might think you're working with something boring or not exciting or just Peronia or just a polychaete or just a nematode. But maybe in 10 years or in 15 years or in 100 years, it becomes important. So think of, that's kind of the end game. As well, we talked about backing everything up and where do you keep your notebooks. We talked about this last week, but this is the first half. But what I wanted to do then is I wanted to talk about museum specimens today, following this up. But up until now, any other questions, comments? Yeah, Mari.
Slides. So in my case, like nematodes are.
You can put the slides in a museum, no problem. And the slides are really great because you actually have a space where you can write. Yeah, slides are totally fine. Slides are specimens. Okay. Good question. Anyone else? Yeah, I put the anemone into a museum and he found it in the museum. So one way to also ensure that your specimens have a useful life after you've killed the animal is you put your specimens into a museum. And by doing that, someday, sometime, someone maybe will come and look at your specimen, right? So that's the idea about specimens for sure. That's how he's going to be sun-founded. Good question. Any other questions? Yeah, Stefan. Is it easy to put a specimen in a museum in Okinawa? Yes and no. Right now, there are two main specimens and two main museums and another kind of museum. The two main museums are the Fujukan at the University of the Ryukyus. It is full. It is more than full. There are specimens in the aisles, and they don't have anyone really to look after it. But it is a nice museum, but it's not super well curated. So I would not put any type, we'll talk about that in another class. I would not put a type specimen in the Fujukan. There is the Okinawa Prefectural Museum, which is down in Omoromachi. They do not have so many biological specimens, but they do have funds, and it is a prefectural museum. You might consider putting a type there, but they're more not so much biology people. I think there are some crustaceans there. Nonako-san and Shurashima Foundation have a small collection of octocorals and antipatharians, etc., up at their office. It's a semi-private collection because it's in their office areas. And I wouldn't add more. It's almost private. So in Okinawa, there's not really a good place to put your specimens. However, there is talk that they are going to build a national biodiversity museum in Okinawa, but it keeps getting delayed. So maybe this is in 10 years. If so, great, but we don't know. There is a museum though that you can put type specimens. This is in Tokyo, the National Natural History and Science Museum. Kahaku is the name in Japanese. The main office is actually in Ibaraki, in Tsukuba. I have put many specimens in there. But because it's the National Museum, they don't just want random specimens. They want new species or type specimens. So if you have a lot of specimens right now, we don't really have anywhere to put them. We could put them in Fujukan, but they'll just sit there, right? So it's not a perfect situation. You know, ideally, I think it'd be great for the biology department to make a big room, but at least for all of us, for Koeda Sensei has specimens, but right now there's no perfect answer. We have a room, it's very dirty. There's many specimens in there. Hopefully in the future, there's the Biodiversity Museum. We'll talk more in another class about type specimens, because types are really important. But for your specimens right now, keep them as best you can. And then we will, ideally, in 15 years, when I retire, we put everything into a museum somewhere. But where? Every museum in the world is full. That's the problem. Everyone says museums are important, but no one is funding museums. So yeah, no perfect answer. Good question. Anyone else? Japanese, English? Kenta's okay? Kenta's okay. Okay, but let's talk about museums. Before we go further, who has done work in a museum? Nuria and Roger and Ifena and yeah, Mari says yeah, and Chloe says yeah. Okay, so 50-50. Okay. Some of the stuff, some of the advice I give you is going to be very easy, very obvious, but other stuff, maybe there's some information in there. Okay, museum specimens. Before you go to a museum, okay, for example, for marine animals, where are famous museums? Where are big museums for marine animals? Just give me some names.
in Okinawa.
In the world. Huh? Pardon? Seckenberg in Germany. Smithsonian in Washington, D.C. Natural History Museum in Paris, which is famous for being huge and a disaster at the same time. London Museum. Naturalis in Netherlands is one of the biggest in the world. The one in Tokyo is not bad, actually, to be honest, for marine specimens. So there are Australian Museum. It's also quite famous. There are a lot of museums. Naples has a big museum. Where else? The aquarium there has a, yeah. I think so, yeah. There's specimens in there. Oh, Monaco. Sorry, Monaco. I'm getting confused. Monaco has a big collection, old collection. There are many places in the world that are famous. And maybe you could get funding, right? You could apply inside, oh, I'm going to get money. I'm going to go to Smithsonian, yay. But before you do that, please contact someone. You might go and they're closed. You might go and the manager's on holiday or something. So if you want to go to a museum, every museum homepage has a number or an email or somehow that you can contact these people. And then they also should have, every museum should have a list of what is available. If they don't, the manager can send you a list, like an Excel file or something. So if the information is not public, you could still contact them. You could say, hi, my name is Chloe. I like worms. Send me a list of your worms. And boom, they will. Because museums' jobs are to work with researchers like us. And so they want, most of them want to help you, right? At the same time, when you contact the manager, you might want to say, you might want to explain, what do you want to do? Do you want to look at their specimens? So for example, in corals, right now, like Francesca and Andrew and a lot of people, they're visiting museums and they're getting photographs of the corals because of their skeletons. And they want information on the morphology of the animals. So they're not sampling, they're not destroying, they're just taking the types and they're getting photos. Other people, maybe if you're working on zoantharians, maybe you want a bit of tissue to do some DNA or some next-gen DNA. So you need to say, like, I want to visit, I want to sample a little bit if I can. Sampling from a type, some people say, I've been able to sample from types, but some people are and some museums are strict. So you need to be really clear, really transparent. Who are you talking? What do they have? What do you want to do? And ask them about their rules and regulations. Maybe there's paperwork. Maybe you need the inkan of your sensei in Japan, or there's all sorts of rules. So before you just go to a museum, make sure you've done your homework, right? Then when you go to the museum, of course, this is just like lab meetings and everything else. Make sure you've got your times and schedule. Don't be late, right? Easy stuff. Bring your own toolkit with you. I don't mean necessarily toolkit, dissection kit. The museum may have some equipment that you can use. They will have microscopes. They will have probably trays. They will have maybe stands and things like that, but they might not have knives or tweezers or pointers or things like that. And so each one of you is working on a different group of animals. And so the tools you use are a little bit different, but don't show up to a museum with nothing. What I'm saying is if there's any sort of handheld tools, handheld equipment, also cameras, SD cards, all of that stuff, flashes, maybe they have a setup, maybe they don't. Bring your equipment to get good data. If you're not sure, email them before, but don't assume. Don't assume they have anything. Maybe even bring your lab coat. Some places are very strict about lab coats, right? Some places yes, some places no. Also sandals, right? We wear sandals in the lab here. Most of the world you cannot. You have to wear safety shoes or hard-held shoes. So different museums have very different rules. If you show up in board shorts and sandals and no tools, you do not look very professional. Good for surfing, good for diving, not good for museums. Cameras, notebook. In other words, try to look like you know what you're doing, right? I think this is pretty obvious, but yeah, it's if you don't know, not very good, right? The other thing is museums and taxonomy, just like marine biology, it's a small world. It's a really small world. So if you go to a museum and someone says, yeah, Guichet came to the museum, he was so amazing. He was organized. He had backup cameras. He had this really great flash system. He did a great job. Probably they will tell people at other museums. And so once you go to 1 or 2 museums, and if you do a good job, or you get good data, then people know, and it makes it easier. It's like a happy, not a sayaku saku, not a vicious circle, but the opposite, right? Like good becomes better, right? Any questions here? No? I'm not saying don't wear a suit, right? This is not a job interview, okay? But I'm saying, like there's probably rules about shorts and sandals. You want to be like, it's a workplace and you want to make sure that you're safe, right? And you also want to make sure that you have your own equipment. That's what I'm trying to say. Okay? Okay, cool. So if you're at the museum, usually photographs are totally no problem. Some taxa, maybe there's rules about flash, but usually even then it's okay. But sampling, subsampling, collecting might have a lot of rules. So make sure that you talk with the manager before you sample anything. In other words, always have permission. If you don't know what you're doing, don't do it. Right? Yeah, I didn't talk to them. I'm not sure. I'll just cut this specimen anyway. No way. Make sure you know what you're doing. The other thing is, a lot of the time, depending on your taxa, on your taxon, the museum might have a lot of specimens that no one has identified. This happens to me all the time. I went to Naturalis in Netherlands for 1 month. They had 650, 700 specimens. There were 12 that were identified. I didn't know that. I went, they said, oh yeah, we have many zoantharians. And I was like, yeah, I'm going to come and I'm going to choose what I want and I'm going to do work. I spent 1 month identifying. But that's good because now they have a collection with species names and identification. And so if you're a taxonomist, you might go to a museum not only to get information, but to give information. You might end up identifying animals for other people. This is really good. Museums are super happy. Naturalis like, wow, we didn't have anyone who knew these animals. We're so happy. We have more information. So identifying is really, really a good thing you can do in a collection. Probably nematodes, probably polychaetes, octocorals, maybe, echinoderms, not so much. Anemones, maybe, yeah. Zoantharians, absolutely. Feronia, maybe misidentifications, right? But adding identifications, you can add new labels to specimens. If there's a label in a specimen, you can write another label and put on top. Totally fine. You can put species name, identified by Iori Mizukami. So that's okay. But don't remove any old labels. That's the rule. So sometimes you find a specimen with 3 or 4 labels, and you can see the history of who has visited the museum and who's worked on the collection. It's super cool. And you can see who doesn't agree. Right? You can see arguments over time, right? So you never ever remove, you never throw away. You can take out of the jar to take a picture, but you never throw away old labels, but you can add new labels if you want. And then super good advice, photos. Take photos as much as you can of things, because just like underwater, this is your only chance to see that specimen maybe for a long time. Questions on this? Yeah, Kairi.
Pero
追加するときに、その追加した人が書くんですか？
Yes. So if you want to add a new label, you would say, I think this is Holiathera atra. You say Holiathera atra, identified by or var, Kairi Takahashi, and then you write the date. Okay? Kairi wants to try now. Anyone else? Any other questions on this? No? Okay, let's keep going. So when you're done, so let's say you go to a museum, you take some photos, maybe you take a little bit of a subsample or something. There's a few things you can do once you're done that will also help. And this is the same as visiting another country and working with researchers. Of course, you say thanks. You might want to share your findings. If you have new identifications or new information, maybe you could update the Excel file that they gave you. You send it to them. This is what I did. This is what I found. And then many people also, they can contribute information, like in a paper. Maybe together you write a paper. We've done this with many museums in Australia. They have a little bit of knowledge about zoantharians. They send us specimens. We write the paper. They don't know so much about zoantharians, but they know about the deep sea. They know the location. They can give information, background information on the area or the ecosystem or the environment. So this is another way to avoid helicopter science and make good network for science. The final thing is, and this, sorry, the order is a bit random, but also depositing. I talked during specimen collection, maybe you put your specimens into museum. That's called depositing a specimen, right? So, and Stefan asked, are there any good museums in Okinawa? So the first thing you do is if you have a list of specimens or something, you should find the person to contact. You should email them or contact them, phone them. Hello, my name is Stefan de Palmas. I have 900 Samakora specimens. Can I deposit them in your collection? And the museum might say, yes, that'd be great, or no, we're full, or something in between, right? The smaller your specimen is, the more chance you have. Maddie, your slides, probably no problem. Polychaete's pretty small. Actually, all of us pretty small. Giant sea cucumbers, maybe no, maybe difficult, right? But definitely make sure that you've got an okay, because museums often will say no. Then when you send, you probably have to send by shipping, international shipping or domestic shipping. Nuria just wrote the international shipping guide, but shipping or sending specimens is also, there's a lot of problems that can happen, especially with Nagoya and with CITES. So make sure that the labels are very clear. Make sure the labels in your specimens are very clear, or type them or print them, right? Labels are easy to read. If there's a type, and we're going to talk about this more next class, a holotype, or a paratype, or a neotype, or a syntype, all of these types as well, you should write very clearly somewhere where they are, because specimens are important, but type specimens are incredibly important. The type specimen is the species. That's what we're going to talk about next class. Wow, went pretty fast today. This was all that I had ready for today, but still 40 minutes, not bad. But any questions on this, on sending specimens? Okay, so up until today, we've done notebooks, we've done data. What else did we do last week? Was that it? Notebooks, data, identification and taxonomy differences, right? Identifying versus taxonomy. And then this week we've talked about collecting and shipping and museums. So this is kind of all the groundwork for describing a species, right? So what I want to do next week, probably the same 40 minutes, 1 hour, probably 2, maybe 3 classes, we're going to talk about describing species. We're going to talk about ICZN, which is the International Code of Zoological Nomenclature. And then we'll also talk about a few other things, some of the rules of ICZN as well. So we're starting to get into really dense, really hard stuff, but really important. But for this, anything, questions last week, this week, Mari? Yeah,
Say, I mean,
yeah,
you can go to a museum and didn't buy something or.
yeah.
How exactly does it work? I mean, I don't really trust anybody. Just like.
No, so you're correct. If you email them and just say, hi, my name is Mari, I want to come look at your nematodes. They would be like, but you say, hi, my name is Marilyn Carletti. I'm a PhD student studying nematodes at the university, blah, blah, blah. Here's a PDF of my paper. Or if you have a paper, I'd like to come to your museum to look at your specimens. Da, 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 da. Then they'll probably say usually yes, if you're a grad student. You're right. Not anyone can come see a collection. The collections are protected, but they're not private. It's a public collection, but the managers are the gatekeepers. They want to make sure that what you're going to do is okay and that you're not going to destroy anything. So that's why also you need to have your own tools. You need to be very clear about communication. I think for PhD students or postdocs, usually, especially if you're with a lab that's doing taxonomy, it's usually okay. You'll usually get permission to go. That said, if you go and cause problems, then it'd be difficult for your Kohai, right? But usually it's something like that. Also, like if you're in a lab, you know, it might be cool to CC your supervisor or maybe someone who knows them. Like I know people at a lot of museums, but maybe some other people, like maybe Stefan knows someone at the National Taiwan Museum or something. And then they might say, okay, but they might email Stefan and say, yo, is this Marilyn, is she cool? And somebody, yeah, she's cool, and boom, right? So there's also that word of mouth thing too. But I think you'd be able to go and look at things, no problem. Other questions? That's a good one. Anything? Daijoubu? Stefan? Yeah.
Data, you need to share the protocol, you need to share the sample with the country where you are.
Basically, with the Nagoya Protocol, if you have a good relationship, if you have the permits and you have a good relationship with the people in the other country, there should be no problem. Should be no problem. Okay, Gio, I see her. I'll get to you next, Gio. So the number one thing for international, the number one advice I can give is have a good relationship with the other country, with the people in the other country. That's the number one thing. On top of that, having a good relationship is good. People sometimes change. Usually they don't. Usually good people are good and bad people are bad, right? Whatever. So if you have a good relationship, that's good. But maybe the person leaves, maybe they get a different job or something, or maybe they quit and start selling coffee, or who knows, right? So the other thing you can do, there's two things here. There's an MTA and an MOU. An MOU is a memorandum of understanding. This is something that usually two universities or two institutes, like a museum or a university, that you sign. And it's basically an agreement that you promise to work together on some kind of research. So an MOU would be like, for example, Stefan, if you are doing research on Samacorin zoanthus and you have a collaborator in Guam who's doing the same, you could sign an MOU to promise that we promise to work together and write papers together and da-da-da-da-da. So an MOU is like a cooperation agreement. And so that protects you if later they change their mind. It also protects you when you go through customs, when you visit other countries, because you can show, oh, look, no, I'm working with this university. Here's my evidence, right? An MTA is a little bit different. It's a material transfer agreement. Basically, an MTA is like a contract that you promise to send something to someone else, or they promise to send something to you. I have an MTA with Pikrik in Palau. And I also have an MTA with Kaust in Saudi Arabia. And this means that if I go to Saudi Arabia and I collect and I work with them, they can send to us, and we have paperwork saying we agree to share bioresources, biodiversity bioresources. So I would say, if you're going to protect yourself from Nagoya and from CITES, the best thing you can do is be friendly and be good with other people. But the number 2 thing is one of these. This you cannot do by yourself. This has to go through me. It's either faculty or university level. These take 3 months to a couple years to complete. They're not fast. Lots of paperwork. But worth it. Especially if you're going somewhere once, I wouldn't worry so much. If you just visit somewhere once and you're done, have a good relationship, maybe a simple MOU or a contract and some letters, like Vale, when Vale visited, we don't have this. I wrote her a letter. These are specimens, they're from Japan, they're legally collected, not CITES, everything's okay. But if you, like I've been to Kaust 5 or 6 times, now Chloe, Iori have been there. Maybe in the future, maybe more people can go. Because we have a long-term collaboration, we have MTA. Does that answer your question? Yeah, okay. Anything else? Ju. Let's see, Ju has a question in the chat. I'll go to the chat. I have hundreds. We've got 2 from Ju. Okay, let's go back. Question number 1. Let's go to question number 1. Have you had issues before with museums, like miscommunication with managers, both when visiting and depositing or borrowing specimens? I have not had an issue with museums. I've never had an issue with miscommunication or with visiting and depositing or borrowing. I've never had an issue. I do know people that have had problems, and it does happen. The problems are usually that one side or the other. Kurt's class just finished. No worries, it's not his fault. Hawaii. So there have been a few cases where like a researcher lost a specimen or something, but yeah, usually not so much of a problem. The next question, I have hundreds of samples and will probably deposit it before publication. Okay, hundreds of samples deposited before publication. I don't think we can deposit those anywhere. No, but you don't throw them away. You keep them, and we hope that there's a biodiversity museum or something in the future. But in your paper, you should at least give the specimen numbers and where they're being kept. Even if it's not taxonomy, if you say the 952 specimens are kept in Ju Lawless's private collection at the University of the UQs under specimen numbers, blah, 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 to blah, blah, blah. At the very least, someone can read that and they can email Ju. And if it's 10 years, Ju says, oh, yes, now those specimens are in this museum, or, oh, yes, I have them with me, or da, 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 da. Once you have specimens, you keep them or you give them to a museum, but you never throw them away. That's why our specimen room is full of specimens. Yes. Okay. Oh, Ju, next question.
I remember Emmeline needed to deposit her sample for her reproduction study, or maybe I'm wrong.
So no, if Emmeline deposits, like we can deposit, then we deposited some for Gishe as well. We deposited like 2 or 3 or 6 specimens or something in Fujukan, the University of the Ryukyu's museum. If you have a few specimens, we did some Kalinura as well. If you have a few specimens, If you have a few specimens, Fujukan is okay. But if you have hundreds and hundreds and hundreds, I wouldn't. The other thing to consider, we'll talk about this more when we talk about types, but a species, if a species is well described, that species is described forever, right? So you can go to the museum in Paris, for example, and you can look at specimens from the 1700s. You can go to Naturalis and they have specimens from 1802 in there. What this means is when you choose a museum for something like a type, you need to choose a museum that you think is in a stable location that will always be there. Earthquakes, civil war, fires. Museum just burnt down in Brazil, what, 2-3 years ago, right? Part of a museum. Things happen. Germany's museums were bombed in World War II. A lot of type specimens are gone. So, and this is beyond human comprehension. What will the world look like in 40 years, in 200 years? We don't know, but as much as possible, you want to deposit your museum, your specimens in museums that are well funded, that are in stable, logical places. So usually when I describe a species, I put one in Tokyo, I put one in the Smithsonian, and I put one in Naturalis. And that way, there's one in Europe for people to look at, paratype. There's a paratype in Smithsonian for people to look at, and the holotype's in Tokyo, which is the country where it was found, basically. Kurt.
Sorry, I'm late, Sensei. Can you hear
No
me?
worries, no worries.
I was just
Hey,
wondering.
thanks for showing up.
We're just done with the classes anyway. I was just wondering, you were talking about holotype specimens. Because back in the Philippines, there is a centralized rule that for holotype specimen, it should be deposited in the National Museum of the Philippines. Is there a rule here also here in Japan that there is a centralized museum? Maybe you've discussed it earlier, so I wasn't able to, catch up.
Okay, I'm going to talk more about types in the next class, but in general, Japan has no rules. However, in taxonomy, generally speaking, the best thing to do is to deposit a type in the museum of the country where you found it. But sometimes that goes against what I just said, which is having a stable museum. So you want to follow the rules of the country as best you can, but you also want to protect your type into the future as best you can. So it can get very, very difficult. Philippines National Museum, is it well funded? Sure.
I think you can ask Joseph better than me.
Jew. Okay, Jew.
I mean Joseph.
Joseph the sponge
okay.
guy.
So
Joseph Comendador.
right? Philippines, Brazil, Indonesia. These 3 countries have very strict rules on biodiversity. But the Brazilian museum just half burnt down part of it last year, 3/4. A lot of it burnt down. So what do you do when you have national interest and rules versus longevity and safety? It's a tough decision. I've never had to deal with that yet. I've described species from Palau, but the Palau Museum doesn't want marine specimens, so I put them in Tokyo. And it was a Japan-Palau collaborative research project, so that was okay. But other species I described from the Dutch Caribbean, they're in Naturalis in the Netherlands. I have a few species I described from Canada. They're in the British Columbia Museum in Victoria. So you want to do as best you can, but sometimes it gets tricky. But thanks to CITES and thanks to Nagoya Protocol, the chances of us describing a species from Indonesia, the Philippines, and Brazil right now is pretty low, unfortunately, unless you happen to be a national from that country, like Roger from Brazil, for example. So the problem with countries that have very strict rules, the good thing is they're protecting their biodiversity. The problem is they're making science suffer. And so you need to find that happy medium, right? But I don't know. Japan has no rules, no. Sorry, long answer to a short question, Kurt. Thank you.
Thank you, Sensei.
Yeah, she's only got 1 species. Exactly. So Ju, she deposited 1 single species or 1 or 2 specimens into the museum just as vouchers, basically. Okay, any other questions? Wow, amazing. This class is going to be an hour after all. Oh, Ju, go ahead.
I would just like to make a comment from someone from the Philippines.
Yeah,
Just for everyone to know, I think it's collecting specimens there is very doable, but the critical part is finding a collaborator, but it's really doable.
yeah.
Just encourage everyone who's planning to collect theirs in the future or
I think
something.
I don't want to speak badly of Indonesia, but I think it is much more popular or not much more possible than Indonesia. Indonesia has rules where if you can do the analysis in Indonesia, you must do the analysis in Indonesia. So I don't think the Philippines has that. From talking with some international people, like Ju says, you need to have a good relationship. There's a nudibranch person, Terry Gostliner. He's described many species from the Philippines, and he has very good relations with most of the local people there. So if you want to focus on a country or something, develop those human relationships, 100%. Cool, Ju. Yoroshiku onegaishimasu. Yuri's laughing. No, but I think it's important, just like in Japan, right? So, Kurt, question.
We're talking about depositing like specimens, right, Sensei? Because
Yeah.
I've encountered before a reviewer, because I just want to ask on a perspective on what like specimen, right? And you want to deposit it. So in my case, coral. So I only took fragments. So we presented in the paper, and then one of the reviewer mentioned that this is not a specimen. So
Why
he or
not?
she mentioned that you need the entire colony, not just the fragment. So I just want to ask about your comment on that one.
Well, no, that depends. That's taxa to taxa. But a
Yeah.
fragment of a colony is a specimen. A single polyp of a colony is a specimen. A single slide of the nidae of a specimen is a specimen. A specimen doesn't have to be a whole colony. The thing is you just have to be very clear when you're writing a paper what you did. If you write, I collected a small branch of each colony, and these samples or these specimens are that's a specimen. There's nothing wrong with that. The reviewer is mistaken in my view.
So maybe I think his or her point is the danger of like, so like from this one colony, for instance, like I collected one fragment, I deposited it in the museum, and then there's this another, like probably scientist, then collected another specimen from the same colony. So there are like two specimens now. Like, I don't know.
No, but how do you know it's the same colony?
I mean, just like it's like a hypothetical.
I mean,
So maybe,
hypothetically, you could run into these problems. That's why you put as best information as you can on the label, right? But the flip side is, because someone else might collect it, you must collect the whole colony. It's nonsense. Does that mean we should just go kill the last rhinoceros because we want to make sure that no one makes a mistake? No way, right?
that makes sense.
I think the main thing is you want to be clear. Like if you write a paper, not only on what your collection, but on what you collected. If it's a colony and you didn't collect the whole colony, you need to say, I collected part. That's why in situ photos are also important, right? You could even show which part you collected in a photo, right?
Right.
Is that okay, Kurt?
Yeah. Thank you very much.
Okay, Guichet, we've opened a can of worms.
Is there a way to like not to track or follow or kind of like register the species without collecting?
So some people have argued that there should be ways to have photographs as holotypes. This has been a huge controversy in taxonomy recently, particularly with jellyfish and ctenophores, because these are animals that do not preserve well, especially ctenophores. You put them in ethanol, they disintegrate, they disappear. You freeze them and you end up with like a tissue with snot and nothing else, right? So you cannot really preserve some ctenophores very well. The authors argue that a good photograph or a 3D photographic, like orthometric rendition of a species should be a type. I think no. I think genetics, I think extracted DNA could be a type, but I don't think a photo could be a type. But you can have photographic records, and I've used those a lot myself. If you have a photo, it's been identified by an expert, and it's a photograph from the field, and you know the location, or there's a GPS on it, you could use that as a record. But I think for a specimen, you could use a photo as a specimen, but yeah, difficult. But it's this huge controversy. Go look up these papers on Google Scholar. It's a really big controversy amongst ctenophore and jellyfish people in particular. Luckily, looking around at most of us, we work on groups that are pretty easy in that sense. I mean, for Mari, you have to destroy the animal to get DNA, but not so bad. Yeah.
But can you maybe have both, like as you said, DNA and a picture?
Oh yeah, no, that's why you take, ideally, you are taking photographs of every single thing you sample. And so you have, because once you fix or preserve a specimen, it looks different, right? So you need those good photographs when you're going to describe a species as much as possible. Absolutely. Okay. Okay, Mari. Yeah, the problem with nematodes.
Now that I did a lot of, I tried different things, and they're so fast when they dissolve it.
Yes.
I can even take a picture of them and move the diagram to the DNA.
Yeah.
Because they're like so small, and the time I take the picture, they explode, they are destroyed. So
Yeah.
in that case,
Mm.
I have no what I extracted on it.
So when there's a couple ways you could do it. You could, instead of taking pictures, if you're using a microscope with a camera, record the video.
Okay.
So at least you can see it for that 1 second before it disappears or something, right?
But yeah, this tuba is like a line or something. I mean,
Yeah.
I can tell what it is.
So up until now, when people describe nematodes, what do they do? How do they do this? How do they deal with that problem?
That's the question that I'm asking,
Yeah.
because they say that they did both, like they use the same specimen.
Yeah.
both, and
Mm.
like how did you do that?
Hmm.
Like it's okay for big nematodes, but with the small, which
Mm.
which are the most described for this reason probably.
Yeah.
So big animals, no problem.
Yeah.
Same size of body is no problem, but the very small ones, I don't think they are.
So
they did it.
what I would do, honestly, if this is something that you cannot find an answer in any papers, I would email the person on worms, the person in charge of the nematode taxonomy on worms. I would email them that question and say, what do you do? Or maybe they answer, no one does this because it's too problematic. In that case, maybe we can use the genomic DNA as the type specimen.
But what person, like, where do I find
So go to worms,
Yeah.
look up your group of nematodes that you're working on. The name and the email address of that person is on the homepage. I think I talked about it last class. Yeah. Check that out. If you want, CC me and say, because ideally for many of your groups of animals, right, that one of the end goals is to put a name on it, right? It's nice to have a name on something, right? It's like, did my job, right? So just because no one's done it doesn't mean it cannot be done, if that makes sense.
Then
Complex grammar. So I'd email them and let's see what they say.
But do you need a picture for like
That's
to describe or for any publication?
That's next class. Next class, when we talk about species, we're going to open that can of nematodes and talk about it, about pictures and things like that. Good question though. Agus is planning to establish a Mise-like lab in Indonesia someday, probably with a much bigger social media output than us. So we can probably do molecular stuff and analysis in his lab. Yay! Okay, so no, this is and this is something I think that's good, is developing a network. Is we do science with people that we trust, and we do science with people that we want to do science with. Science is human, right? And so I think science is way more fun and way more productive and way easier when you're working with people that you can trust and people that you get along with. So what I would suggest is, yeah, like for example, if Auguste has a Mise lab in the future, if Ju, Emmeline, Jem, and Kurt make like the Philippine Coral Triangle Mafia Mise lab or something in Philippines, there's people around the world, there's a network of people that you can work with. And Auguste is excited. Cool. So, but I mean, I think if you do something like this, that's one of the really good values of this. And so it's not only that we're all in this lab and we're studying at the same time. In the future, maybe in 10 years, like I'll be sitting in a rocking chair, you know, and you guys will be feeding me coffee or something. But and no, I'm still young. But what I mean is you guys might be collaborating on science together in the future, right? And so there's chances here, not only for Okinawa, but for you guys to do work all over the world. And that's one really cool way to get a lot of international work done, is with people that you know and trust. Definitely osusume. Hi. August says yes. Anything else? All good. Oh, Stefan, yeah.
But the rules are right now might change tomorrow. And so like maybe we can deposit DNA in different
So we're going to talk about describing species next week. I will tell you very vaguely right now, the rules are not nearly as bad as you think. It's the scientists that make it a pain in the butt. Number one, Chloe's looking confused. I'll explain this after. Number two is, if the rules are going to change in the future, international collaboration is getting more complex because of rules and blah, 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 CITES, Nagoya. But international collaboration is increasing, and rules and taxonomy is actually getting easier. You might not believe me, but I think it's true. So with that in mind, when we talk about taxonomy next week, I'm going to surprise you guys next week. It's going to be fun. I'm excited. Anything else? No? Okay, today's boot camp's done. Otsukare-sama desu. I will put this up on YouTube after. Thank you everyone online. And see you later. Otsukare-sama. Okay. ちょっと待って。いいよ。今できる。容量が足りない。ちょっと待って。